Hi there, everybody, and welcome to today's Identifying Repurposing Opportunities webinar. My name's Libby, and I'm the Fundraising and Communications Officer at Find Cure, and I'll be hosting today's webinar. Firstly, I just wanted to check that everyone can hear me okay. If you have any technical issues at any time in the webinar, please just let me know by typing your issue into the question box on the control panel, and I'll do my best to help you sort it out. And on that note, we'll be having a question and answer session at any point in the webinar for either of our speakers. You can type a question into the question box and we'll come to it at the end. So the agenda for today, we'll be listening to two very interesting presentations. Firstly from our own Rick Thompson, the Head of Research at Find a Cure, who will be introducing the issues of conventional drug discovery for rare diseases and why drug repurposing is therefore an ideal option for diseases with small patient populations. He will also be introducing our own drug repurposing project and explain how we're hoping to fund it using social impact financing and how this is associated to our open call for drug repurposing projects. The open call is the reason why this webinar has come about, and it's something we'd really love for you to get involved in. We'll then be hearing from Dave Brown, who is the chair at Helix. Helix is a Cambridge-based startup that's focused on repurposing drugs for rare diseases. We're partnering with them on the open call. Dave has 40 years experience in the pharmaceutical industry and was part of the patent for Viagra, which you probably know as one of the most successful examples of drug repurposing. He will be explaining the different approaches that researchers, clinicians and patient groups can use to identify repurposing opportunities. And we're very grateful to Dave for joining us today. And then following that, we'll be having our question and answer session. And as I said before, if you have any questions, do just type them into the question box in the control panel and we'll come to them at the end. And same for any technical difficulties that you have throughout. So with that, I will pass you on to Rick Thompson.
Okay, thank you very much, Libby. Brilliant. Well, thanks for attending, guys. We're really excited to be talking to you all today. As Libby said, my name's Rick, and I am Head of Research here at Find a Cure. And what I want to talk to you today about is our rare repurposing open call, but more specifically how that's come about, the repurposing work we're trying to do here at Find a Cure, and why we believe this is an avenue that's really important for rare diseases and rare disease patient groups to try and get involved in. So we'll start at the obvious beginning, which is to talk a little bit about the background of rare diseases. A rare disease in the EU is defined as a disease that affects 1 in 2,000 people or fewer. So this means in the UK there are about 3.5 million people affected by a rare condition. Across the world that's nearer 350 million. So despite that word rare, if we look at all the conditions collectively, we can see they really are a huge global issue. There are about 7,000 different rare diseases, and this number is actually continuously growing with the rise of genetic diagnoses. We're finding more and more of these conditions, but unfortunately only about 400 of them have licensed treatments. So what you can see there is a huge unmet need for patients to have treatments that can really help them manage and live with these conditions. There are also huge issues with rare diseases around diagnosis. We have very long periods of time, diagnostic odysseys in fact, for patients trying to reach the accurate diagnosis of their condition, and this is a situation that really is not good enough and something we're very keen to change at Find a Cure. What we do have in the rare disease patient population, though, is a group that are exceptionally empowered, that have got power and determination to deliver change for themselves. They're in a situation where patient groups have to take the lead to make sure they get the care that they deserve. And at Find a Cure, we are really keen to ensure we can help empower those patient groups to do that. So that's our primary focus in much of our work, empowering patient groups, professionalizing them and helping them to deliver the type of support to patients that they want to deliver. Another thing at Find a Cure that we recognize is that rare diseases are really fundamental. Rare diseases are playing a fundamental role in the way that we're delivering healthcare now. If you look at patient groups, they are exceptionally active in the rare disease space and trailblazing the type of patient engagement that we're beginning to see across the board in medical research. It's a fundamental change in how we're doing research. Rare diseases are also often used at the cutting edge of scientific discovery, and in many of these new scientific methods like gene therapy or gene editing, rare diseases really are at the heart of it, and we're seeing them trialed in these areas. So they're fundamental to the way we'll treat treat and diagnose medicines in the future. And what this means is that, if you're doing research into a rare condition, you're almost certainly going to impact more common diseases down the line. So subsequently, research in rare conditions is a benefit to everybody. Because of this, we're really keen to encourage the patient groups we help to professionalize or empower to interact with research, to possibly drive forward their own research, or to ensure that something is being done in their rare area. So we work to encourage collaborations between patient groups and the pharmaceutical industry, the biotech industry, academia, research, and in fact other patient groups, all to ensure that they learn from one another and can deliver the best research to patients. So given all this, where are things in the pharma industry right now for delivery of care? Well, essentially, over the past 10, 20 years, there's been a change in the productivity in the pharmaceutical industry. It's not a clear trend, but it is up and down. But what you can see from this graph in the bars is the number of drugs that are approved year on year from 96 through to 2013. And we're seeing a general trend to decline in the number of drugs approved. With the green bar, we can see the average research spend for developing new treatments. And this has been going up. It's plateaued since around 2007, but clearly there's an increase in the cost to develop drugs and a decrease in the number that we're producing, which suggests some level of inefficiency. It hints at the need for some new way to drive the production of new treatments for all conditions, not just rare conditions. Now, if we look at rare conditions specifically, when you have these high development costs, which are generally uniform across the pharma space, combined with a low patient population for a given rare disease, you tend to find that you're going to drive the price of the treatment up. Pharmaceutical companies feel they have to charge more for their drug when it reaches market in order to make back their research investment, which is fully understandable. So if we look at this graph taken from a recent report, what we can see is a comparison of orphan drug prices, those are drugs for rare diseases, to non-orphan drug prices, 2012 through 2016. And what you'll see is a really big disparity. We've got non-orphan drug prices in 2016 to be around $27,000 per patient per year. And if we look at the orphan price, this is nearer the $140,000 per patient per year. So a very large difference there that we need to be aware of and account for. So overall then, the situation in rare diseases can be summarized like this. We've got an average cost for developing a new drug around the $1.6 billion mark, which is pretty high. The number of rare diseases is estimated at about 7,000, which gives us a rough estimate of a budget of $11.2 trillion required to deliver treatments for all of these conditions using conventional drug discovery approaches. This is something that's an exceptionally huge amount of money, and not something that's likely to happen very quickly. So what we need is something that's faster and cheaper to help us deliver these drugs to patients more quickly, particularly in the rare space. And we believe, and I'm sure Helix will agree, that repurposing is a potential solution to this problem. So that's why drug repurposing really falls into something that Find a Cure are focusing on. As part of our aim to drive collaborations, we're looking to develop our own drug repurposing platform. So what is drug repurposing? Well, at its most basic level, it can be likened to recycling. So it's the act of taking a drug that's approved for one patient population to treat their condition and demonstrating it can be used effectively to treat a completely different patient population or a different condition. We want to do that using clinical trials. We want to provide scientific evidence to support that use. There are lots of potential benefits of drug repurposing, and those essentially boil down to the idea that it's faster and cheaper. And this really makes it good for rare diseases when we've already got issues around the difficulties with numbers of patients, making it hard sometimes to understand the basic science of the condition. If we can make things easier for ourselves in this space, it can only be a good thing. So one of the first reasons it's a benefit is that we are eliminating the need for this de novo drug discovery step. So this is a step where we have to identify completely new chemical entities, and this can take a huge amount of time, money to identify things that are safe and useful for human use. Secondly, we know a huge amount about things that we already are aware of. We know something about the safety profile and the side effects of existing drugs. If we're working with a drug that's already generic, so a drug that has lost its intellectual property and is possibly used for many different conditions, this will mean we've got a long history of human use, and we have a history of its use in different populations, maybe different patient populations, maybe in adult groups, maybe in pediatric groups, and all of this tells us far more about its safety and efficacy. What this means is that if we're doing drug repurposing, we can potentially therefore have a reduced requirement for early stage clinical trials. This again can speed the process of drug development and make it less expensive. with drugs that we know, we also know how they work. We know something about their pathways of action. And using this knowledge, we can identify drugs that can potentially benefit a specific patient population through that information. So we might know that 2 different diseases work on the same pathway. If one drug works there, it could work for both those conditions. This allows us to intelligently select potential candidates for repurposing. Again, speeding the process and reducing the cost. Now, as I mentioned, generic drugs are something that have a wealth of data and should therefore be quite appealing for us to look into repurposing. However, there are issues with doing that in the industry, and that's mainly because it's hard to secure intellectual property on these generics. Their patents have already expired. The main route people can use is some level of protection from orphan drug designation, but also the use of mode of use patents. These are patents that say, I will use this drug for this condition. That can work and can be a successful strategy, but it can be hard to defend these patents because it's possible that other formulations or other versions of the same drug might be available off-label, and these can be prescribed at a different price. Basically, therefore, it's hard for the pharmaceutical company to recoup its investment. And this means it's more difficult for them to really work in this generic drug repurposing space, so we don't see as much action in this area. So unfortunately, we get this fusion of high developmental costs, and that's combined with these small, fragmented, rare disease patient populations, and we see nothing much happening, my tumbleweed of financial inviability. So that's something we're keen to change. And we think patient groups are a particular division of the rare disease community can actually drive this forward. So here we have an example of this, which is the AKU Society or the Alkaptonuria Society, who are a patient organisation who are helping to drive research forward in their condition. So alkaptonuria is otherwise known as black bone disease, and it's caused by the absence of an enzyme that degrades homogentisic acid in the body. So homogentisic acid, or HGA, it tends to build up, and what this does is causes bones and cartilage to turn black and making them much more brittle. So this leads to a very early onset form of osteoarthritis, which can be very painful and lead to multiple joint replacements for the patients. Now, the AKU Society, along with one of their trustees and a clinician, Professor Ranganath, have worked to identify a drug which is used for another condition, tyrosinemia type 1, that works on the same pathway as alkaptonuria. And what this means is they're able to try and repurpose it into alkaptonuria itself. They secured a huge pot of funding from the European Union to run clinical trials called the Developer Cure trial to help prove that nitisinone can function as a repurposed agent in alkaptonuria. So it's been working really well. They've completed their phase 2 clinical trial and are now well into their phase 3 trial to show that nitisinone is an effective or efficacious drug for alkaptonuria. And we really think this is a model that we want to see replicated across the rare disease space for patient groups. Now, unfortunately, not all of us can access delightfully large pots of European funding, so we have to have ideas of different sources of funding to drive our research forward. And one of these routes could be to look at Cure Accelerator. So Cure Accelerator is an online platform that's available to promote and help fund repurposing research. So this is run by the charity Cures Within Reach, who are also partnering with us on our Rare Repurposing Open Call. And it's simply a forum where people with repurposing ideas can try and meet people who are looking to fund repurposing projects. So to give you a little bit of background on the success of the Cure Accelerator, it's been running for about 1.5 years, and it currently has over 1,000 different active users. These users are spread across about 20 different countries, and in January alone, 3 different funding opportunities were posted on the Cure Accelerator platform. Over 130 different projects are present there currently. These might be projects looking for repurposing funding or calls for repurposing projects themselves. And it's been relatively successful in that almost $1 million of funding has been allocated to repurposing research already during that time. And this is spread across 19 different projects in that 16-month period. So this is a new way to try and match up people with funding to help drive research forward, and this might be in preclinical space or in the clinical space. So it's certainly a website worth investigating if you happen to represent a group who are looking to secure funding to help drive repurposing research forward. Something else to look at for funding is what we're working on at Find a Cure, which is the use of social finance. So social finance is a different way of approaching money and investment. In a normal investment, the primary aim is to secure an economic or financial return. In a socially minded investment, the investor is also interested in the benefit to society that their investment can deliver. And this is often the primary outcome that they're focused on from their investment itself. So socially financed projects, therefore, tend to be delivered or completed by charities or third sector organisations. And they'll aim to provide some kind of intervention, something that will deliver some form of social change. And generally, they receive some form of government funding or payment based on the success of the change that they deliver. So this is generally referred to as a payment by results mechanism. Now, what we're looking at is a social impact bond as a form of social finance. Now, the social impact bond has been used previously to reduce reoffending rates in prisons and to reduce truancy in schools. And it's something that's been seen increasingly, particularly in the US and the UK. The first one was set up in Peterborough around 2004. What we think is we can use this model to help fund generic drug repurposing. So the question then becomes, how does that work? Well, to have a social impact bond, what you need are three major things: a social problem, an intervention that you can deliver, and a measure of success to tell you how effective your intervention has been. In our case, the social problem is clear. There are a large number of rare disease patients out there with no treatment and little hope of ever securing a treatment. And in many cases, these have a high burden of care to manage those symptoms of their condition. The intervention, we believe that we can find and identify generic drugs that have the potential to treat rare diseases and to run clinical trials to prove their effect and efficacy. In this case, our success measure would therefore be the number of rare disease patients that receive a repurposed treatment, and probably more pertinently, the number of patients that are benefiting from that treatment. So the big question there is, where is the money? How can we use this model to actually fund research? And the answer really is in the reduced burden of care. We believe in many cases there are untreated conditions that are creating quite a high cost into healthcare systems. And if we can deliver a successful treatment into that system, we can reduce their spend, that burden of care, and therefore save money. And those savings could be the money we want to try and unlock in a social impact bond model. So how does it work? Obviously, when you model, you need a fancy diagram to explain it, and this is here. Here we can see the aim of the bond is to attract money from investors. They're going to put their private investment into the bond with the aim of delivering this social change and possibly securing some money back as well. What Findicure are going to do is draw funds from the social impact bonds. What we want to do with that is use that to fund a portfolio of phase 2 clinical trials in generic drug repurposing research. We want to identify and focus on generic drug repurposing projects that focus on conditions that have a high burden of care to the National Health Service in the UK. The idea is then, if we can deliver a generic treatment in these areas, it will be delivered in the NHS at low cost, and this will lead to savings in the NHS itself. So we're going to save them money. What we need to do is access a proportion of that money that the NHS saves and get an agreement that they will pay that money back into the social impact bond after they've made the saving. With this, we can then deliver returns back to our investors and hopefully, as you can see on point 6, have money left over to pay back into the social impact bond cycle to fund further clinical trials, further repurposing research. So the key thing here really is we're generating returns from healthcare savings. We're not generating returns from a high-priced drug. And this is the model we're trying to develop in collaboration with the NHS. So we've not just got a fancy figure, we've gone away and tried to work out whether this can actually work by conducting health economic studies in 3 different rare diseases which have specific generic drug repurposing projects at a stage that could be moved to a clinical trial. So those are congenital hyperinsulinism, Wolfram syndrome, and Friedreich's ataxia. What we've done is built cost of illness models to try and understand what it currently costs the NHS today to treat these conditions. And none of these have obvious treatments, they're all just managing the symptoms or finding ways to help the patients cope with the conditions. And as you can see, in all cases, we've got a reasonably high burden of care. For example, in Wolfram syndrome, the price looks not huge to the NHS, it's less than $1 million, but we're talking about a number of patients that's in the region of 64 across the UK. So a very small patient number leading to a cost to the NHS of just short of £1 million a year. What we looked at then is how a repurposed treatment could affect that budget. How would delivering a current generic drug at the current generic price lower the NHS spend based on how we believe it will improve patient health. What we've shown in all cases that it would save money. These are figures across a 5-year period, and what you can see is we're saving reasonably significant amounts of money to the NHS using this scheme. And in this, they're paying for delivering the drug they're not currently delivering and saving money through improving patient health, which I think we can all agree is a positive sign. Again, these aren't vast numbers to the NHS budget, but they're the types of amount of money that could make a real difference to a rare disease patient group or researcher if they had that to use to fund repurposing research itself. So what we believe is our proof of concept study has shown this model can work, and what we need to go out for now is to find more examples, ideally those that are possibly leading to even bigger savings, to try and help us move this model forward with the NHS. And that's really what led us to the Rare Repurposing Open Call. We are looking for projects, and we think the best way to do that is to make a call to the rare community to look for repurposing projects. And to do this, we've come together with two partners, Cures Within Reach, who are a charity focused on drug repurposing in the US, and themselves came up with the Cure Accelerator platform, and Helix, a Cambridge-based startup who are focusing specifically on repurposing drugs for rare diseases. The Rare Repurposing Open Call itself launched on the 28th of February, that's Rare Disease Day, and we're running it for a 3-month period to the end of May. And we're really seeing this as a chance for researchers, clinicians, patient groups, or even people in industry to try and identify the repurposing projects they have, to highlight the need for the support for generic drug repurposing for the rare diseases, and hopefully to find ways to advance these projects and deliver some meaningful change to the patients that need them. So what are the core aims of this call then? The first and primary aim is just to identify repurposing projects for rare diseases that meet these 3 criteria, that are scientifically robust, that can potentially be impactful to patients, that are really affecting the condition in a way that's meaningful to the patient groups themselves, and to try and ensure that we're targeting those conditions that are struggling to secure the necessary funding to move things forward to a clinical trial. And that's as likely to be because it lacks an obvious route for financial viability. And that's something we'd like to address. Secondly, we want to really just promote repurposing as a valid route for rare diseases in general, whether that's to researchers, clinicians, or patient groups. And we really want to see that repurposing can deliver meaningful treatments to patients. So we're aiming to build a body of evidence to support the idea that there are repurposing ideas out there, and they're struggling to secure the funding they need to reach clinic. And we just want to make sure that we're introducing this concept to anyone who's interested in finding treatments for rare diseases so they can explore this and hopefully drive things forward to their in their community. So why should patient groups or researchers get involved? Well, we think there are a number of different possible outcomes which range from things that are essentially altruistic, helping your community, to things that could deliver real meaningful change directly into your project. So generally speaking, we think this call is a way to help encourage the use of repurposing to a whole range of different groups, and a way to help develop new funding mechanism for rare diseases in general. So it can help us to deliver this social impact bond funding model, and it can hopefully help to encourage people to fund repurposing in the future. More specifically though, we want to help the specific projects that come through. So we want to secure support in developing health economic models for repurposing projects. This is something that Find a Cure have worked on quite a lot and can provide really helpful supportive evidence to drive to show the value of the repurposing research, to show it can save money, not just deliver meaningful treatments to patients. We also want to secure support and advice to develop the study from our project collaborators. So, you know, the best projects will be looked at by teams at Cure Accelerator, by potentially funders at Cure Accelerator, by the team at Helix, and hopefully we can use this to help identify different ways and obvious next steps in the project to move them forward. Because anything you propose will be on Cure Accelerator, it has the potential to be viewed by a whole range of different funders, and that could really be a great way to attract funding into your project and hopefully move the research itself forward. And finally, if we can really develop this social impact bond model, the ultimate aim is to identify projects that are perfectly suited for it and to be able to deliver funding for a clinical trial in the social impact model itself, and really blaze a trail for a completely new way to finance generic drug repurposing for rare diseases. So hopefully there's a whole range of things there which speak to you and make you want to get involved with this repurposing project. If so, who can take part? Well, anyone and everyone really is the short answer. Research and clinicians are an obvious target. They themselves can very easily sign up to Cure Accelerator and respond to our request for proposals there. It's a simple process. You just have to get a sign up to log in, and by searching Rare Repurposing Open Call, you will easily find the call. We think companies are also a logical group to look at this. A number of companies out there might have ideas for generic drug repurposing projects that just aren't financially viable, that they don't ever believe they're going to take forward, but could have real potential to help the patient groups themselves. And we'd like to see companies, looking to submit these ideas to the core themselves, and hopefully this could be a way to drive their project forward. If they feel there is commercial potential in this idea, there's also an option within the submission process to keep things very private, so we can look at different ways to develop it alongside them in a collaborative manner. So definitely companies should look at this route as well. Thirdly, and finally, patient groups really can have an impact in this area, as we highlighted with the Accelerator Society earlier. Patient groups are able to sign up to Cure Accelerator and view the request for proposals, but on the Cure Accelerator platform itself, there is a need to have a particular research status to submit a proposal. So in order to get around that, you could either work with your existing research boards, scientific advisors, to get a proposal there. If you're not in that position, then we really would encourage you to contact FinderCure directly with your idea. We can send you the questions themselves that are involved in the request for proposal form, and you can fill that out yourself, and we can think about ways we can help you advance that project. So, given all this, what's an ideal project? And that's an important thing for you to understand. So I think first up, there are criteria we're looking for in ideal repurposing projects that are probably shared by all the collaborators. We're aiming to repurpose a generic drug for rare diseases. What we want to do is make sure this has got scientific robusticity, if you like. We're looking that the candidate drug is supported by some level of preclinical data. That would be fantastic because it gives us confidence moving forward into a clinical stage. We're also, in our ideal world, would love to see some in-human case studies, some evidence or understanding of how this drug could potentially affect the symptoms in patients. What we want to know and believe is that there's strong evidence that a repurposed drug, would have a disease-modifying effect, and that would deliver significant patient benefit. So it's not good enough just to say it might change the levels of this chemical. We need to know how that chemical really will impact patient lives. That's something that's crucial to find a cure, and I think very important in any drug development project. And finally, it's important to have metrics and biomarkers that we could use potentially to assess the impact of the drug in a clinical trial, a simple way to measure it. Again, these are the things we're looking for in an ideal world, and all of these are really going to help us drive these projects forward. Secondly, there's another set of ideals that we have which are more tied to our desire to find projects that fit into our social impact bond model. We're very open to submissions that don't necessarily fit into this, because this is the aim of all the different collaborators, is to find new projects that we can drive forward. But if you're looking to move forward with the SIBA model, we'd ideally need a UK collaborator. This is something that's going to help us interact with the NHS, and that's where we're looking to make savings initially. We really need to be able to reduce the healthcare spend on the condition. So for this, we want to have some idea of the current spend, if possible, and the evidence of supporting that. Ideally, we'd like to have something we could start in 2018. And we really do want to see a connection between proposing research institutions and a patient group themselves. Firstly, this means that the patient perspective has been taken into account, and secondly, it gives us confidence that there's a chance to access patients to deliver a clinical trial itself, and all of these will help us deliver something more effectively within our social impact bond model. So as I said, the submission process, we've tried to design it to be as straightforward as possible. The form is available to any member of Cure Accelerator and very easy to access, and can be downloaded onto your computer to share with collaborators or clinicians that you might work with. We've designed the form to identify the most suitable projects for the social impact bond, and that's really through necessity. This is probably the most stringent set of rules we have to have, and we have to be able to assess projects very clearly to see if they fit with it. But that doesn't mean things that don't necessarily nest perfectly in the social impact bond shouldn't be submitted. We really want to welcome all kinds of submissions that could potentially drive generic repurposing research forward. So if you're looking at the form and feeling like you don't quite fit the criteria perfectly, please don't worry, submit anyway. We want to build this case, this evidence that there are repurposing projects out there. The other things to really appreciate is we're trying to balance a kind of ease of submission in the form compared to the need to secure the details that allow us to assess how the project is going to be delivered. So it is structured as if you're applying for research funding. It should be relatively familiar to any researcher or academic who you have working on the project. A basic budget is required, but this is really very stripped back. I think it's probably 5 or 6 different points. You have to give a summary of the costs, and the information we're looking for on the cost of treatment currently is quite important as well for the social impact bond side of things. But if you have any information at this and can provide evidence to support your estimates, that's really appreciated. Basically, if you've got any issues with the submission, please do get in touch. We want this to be as smooth as possible, and we're very keen to work with you to make sure any ideas you have can be delivered smoothly. Okay, so you're sitting there and thinking, but we don't have a repurposing project right now. What are we going to do? And the point is, this is why we're here today. Our next presentation is going to be about how you can come up with new repurposing ideas. And there are a huge number of different ways to do that now. There are many companies and groups out there trying to work to identify different repurposing opportunities, using drug screening approaches, using data mining approaches, or using some of these new exciting genomic or transcriptomic approaches to identify different types of repurposing opportunities in rare diseases. So if you're interested in doing this, if you're interested in trying to find an idea, reach out to researchers that you might work with, members on your scientific advisory board if you are a patient group, or different companies to try and form your own collaborations. And hopefully then you can move into this repurposing space and have a chance to identify something that could really help the patient population. If you've got the tiniest idea, the tiniest nugget of a concept, please do share it with us, because we want to just be able to show that ideas are there that need moving forward, and this is something that can be really beneficial to the patient population. Okay, that is all I wanted to say. Thank you very much for sticking with us and listening. I hope it's been useful and interesting, and I'm pretty sure that our next presentation will give you some great ideas and insights into how we can identify new repurposing opportunities as rare disease patients.
Thank you very much to Rick for that very interesting and useful presentation. We're now going to be passing on to Dave Brown, who, as I introduced before, is the chairman of Helix. He will be explaining the different approaches that you can use to identify repurposing opportunities.
Hello, everybody. Thank you to Rick and Libby. So as they introduced me, I'm chairman of Helix, and I have with me the CEO of Helix, Tim Williams. Together we formed the company 3 years ago. And also Ramila Patel, whose email address you'll see on the front slide, and Ramila is commercial head for the company. Okay, so if we go on to the second slide. I've been involved in repurposing myself. I was just calculating it for over 30 years now, initially almost by accident, but in the last 10 years or so more deliberately. The various roles I have at the moment, apart from chairman of Helix, I'm chairman of Babram Institute Enterprise. So Babram Institute is a world-class biology research institute on a beautiful campus just south of Cambridge, with about 300 world-class researchers there. It's a postgraduate biology department at the University of Cambridge, and I chair their commercialization arm called Babram Institute Enterprise. Also, I'm a trustee or director of Antibiotic Research UK, where we're focusing on repurposing very heavily, trying to beat the antibiotic resistance problem. Early in my career, I've worked with 4 of the top 10 pharmaceutical companies, what's now AstraZeneca, GlaxoSmithKline, Pfizer, and Roche. And I was head of all of Roche's drug discovery with 2,000 scientists ranging from West Coast USA across to Japan. While I was at Pfizer, I led the team that eventually discovered Viagra. We started out, actually, I think it's well-known story, trying to lower blood pressure, found a profile that looked a little bit like an oral drug for treating angina, so we repurposed for that during the research phase. And then in the clinical phase, when we were running those clinical trials, we hit activity in, actually not patients, in volunteer men, where we got erections unexpectedly and immediately understood the science, so we repurposed it for male erectile dysfunction. So I led that team for 8 years, right through to the point where we discovered the effects in the clinic. Also, we repurposed it for pulmonary arterial hypertension. It's sold as under the name Revatio. It saved many lives, particularly in babies that have immature lungs. Makes a big, big difference to their survival rate. Current focus, as I said, trying to beat antibiotic resistance at Antibiotic Research UK. I'm not going to talk about that today. I'm going to talk about my other focus, which is Helix, which is focused totally on rare diseases. So the next slide, I'm going to talk today about these 5 things. A little bit about Helix in general, where the company's at, drug repurposing, open call. I'll skip over that because actually Rick has done a very good job on that. One of my sort of bugbears about drug discovery over the years has been that the methods are very poor, the failure rate's very high. We heard from Rick these enormous costs. They're driven largely by failure. Very few drug R&D projects get a product to market. And I've analyzed the reasons that the old methods fail. I'm going to present to you the new methods. I do believe that what we're doing at Helix, combining the old with the new, will be far more powerful and will lead to a much better success rate. And I'm going to give you an example at the end of the presentation of one success that we've had recently, the CDKL5. So Helix, transforming the lives of rare disease patients by intelligently matching drug treatments. That's our mission. It's a technology company based here in Cambridge, UK. We've got a full-time team of 12 at the moment, still building the team. We've got a number of external collaborators and consultants, which is just about double the size of that team. We're developing machine learning algorithms based on genomics, transcriptomics, and various other methods, which I'll show you later, focused on repurposing drugs for rare diseases. I do believe myself that actually repurposing is going to be the most productive form of drug discovery in the next 5 to 10 years. We have an enormous opportunity now to find other uses for the many drugs and nutraceuticals which we have available to us. But our particular focus is working with patient groups. We're very patient-centric in a way that the pharmaceutical companies just cannot be. But also we're working with academic groups here in Cambridge and elsewhere. So if we go to the next slide, this is how we work. First, we define the unmet medical need, the therapeutic need. With any drug R&D program, it's essential to understand what the patients actually need. So we work very closely with patient charities to understand the disease, the symptomology, and exactly what kind of profile of a drug would make a big difference to the patients. We then move on to looking at what scientific information is available, particularly looking at the transcriptomic data, in other words, understanding how the expression of the genome in a patient differs from that in somebody who doesn't have the disease. Now, what we find is there's often information available, but it's very poor quality, so we have to curate that information, in other words, assess it very carefully, eliminate databases that really aren't to a standard, that are useful, and use only those that are useful. And if there aren't any useful ones, we can go back to the patient groups and say, well, you know, this is what's needed. Maybe you should fund groups that can obtain the quality of information that's required. Then we go back to the patient groups and agree what the profile would be of a drug that would make a big difference to the patient. So again, working with the patients very carefully at each stage. All those various associations below, APOD, CDKL5, Harvard Medical School, these are the groups that we've worked with recently, both patient groups, academic groups, and some groups which are actually running assays for us as well. I'll skip over the next slide since Rick has already been through that one. So I mentioned that some of the methods currently used for drug discovery have major faults with them. And as I said, this has been a real focus for me, trying to understand why drug discovery has been so difficult. Personally, I've got 3 drugs to market in my career, but it's, you know, most people in R&D spend a whole career and get nothing, which is, you know, quite a waste of lives as well as not helping human health. So up till about the mid-1990s, we used what's called a phenotypic-based approach to developing drugs. This basically meant that you took a chemical molecule, put it in an animal, and see what it does. You measure some kind of output. So for instance, early in my career, I was working in diabetes research and later in cardiovascular research for most of my career. You would take a chemical that you thought might be useful, for instance, in lowering blood glucose. You would inject it into a rat or a mouse and see if it did so. Often you didn't have much of a hypothesis of why it would work. Sometimes they worked, sometimes they didn't. Many of our current drugs were actually invented this way, so it's not a bad method, and particularly in cancer, infectious diseases, when you're trying to kill a bacteria or a virus, yes, this is a way you can approach it. You can just kill those cells and not worry too much about the mechanism, and it can be very effective at discovering drugs. But if you're dealing with complicated diseases, neurological conditions, etc., this really isn't a very good method at all. So the weakness is that even if you discover something you think is useful in animals, you don't know the mechanism. The drug might interact with many targets that you're not aware of, and that can lead to side effects, unwanted effects, and a toxicity risk. And of course, you don't know the mechanism, so when you take it from an animal study into humans, it just may not work, and quite often that was the case. But as I said, that was the main method of drug discovery up to the mid-1990s until we started to get genome information, and it's still used in some diseases at the moment. So once the genome started to appear, and there was quite a big shift in about 1995, 96 in particular. We went to what's called target-based, where you would start with a hypothesis. You would have some idea of what the genetic defect might be, or if not the genetic defect, you would have some idea of what a biochemical target might be in the human body. In other words, you would have a link between a target and a disease. What happened, though, was that this didn't work out as we'd hoped it would. The link between target and disease often proved much poorer than we'd hoped. In other words, our hypothesis wasn't that great. We weren't as good at reducing the side effects as we had hoped. So what we call off-target effects, the side effects, were still there. And again, there was still very poor translation from animal models to humans. So personally, I've spent a lot of time trying to figure, is there a third way? Can we combine these in some way? And I think Helix actually does have a way of taking the best of these two approaches, which is why I think the company is so interesting. The third way of repurposing was actually what we did with Viagra. Initially, we had a hypothesis which was target-based about lowering blood pressure. We then, in rats, found a phenotypic response, so the top approach there, which made it look more like a nitrate for angina. So we took it into patients, angina patients. But then serendipity came in when we found this very powerful effect in male patients. So Viagra went through all three of those at different stages. It's a very good example of repurposing using the different methods. But serendipity really isn't something you can use to solve 7,000 rare diseases. We've got to be a lot smarter than that. So what are the new methods? So really, big data analytics is at the core of this. We now have computational systems and massive databases, which are mostly data rather than information. They're mostly noise, but we write algorithms that sort the information from the noise, basically. And those information databases are based around the disease, the drug, and what actually happens in patients. So we're looking at ontology, related diseases. We're looking at responses that may be similar across diseases. We're looking at omics, genomics, transcriptomics. We're trying to find sometimes a causative gene for many diseases. Of course, there isn't a single causative gene, there are many. We're analyzing data around drug structures, the targets they work on, the mode of action, the side effects they have. And most informative of all, of course, is the patient response, which is what we're really interested in, and whether there's any evidence a drug may give a response that may be useful to a patient, whether the drug is well tolerated, whether it actually targets the organ that we want it to target, and whether the method of delivery of a drug is actually appropriate for that particular patient. So all these are feeding into databases and algorithms that we're analyzing to try and repurpose drugs for rare diseases. So the next slide lists some of the methods that we're using. Omics matching means transcriptomics. And I'm going to show you on the later slide a very simple diagram of how that works. Guilt by association means that sometimes a drug, there may be some indication a drug may give an effect, but it may not be very good. But there may be other drugs that work on similar mechanisms that could be tried. Literature mining, we have very advanced ways of mining the literature that haven't been used before, which are very, very powerful. I'm personally using these a lot now, that give us clues of whether our omics information is pointing in the right direction, or may give us standalone information of whether a drug could be useful. And drug network analysis, I think, is probably the most exciting approaches we have. I'll describe that on a later slide. So this is what the output for our in-house teams looks like. The critical thing here is that we're going from data curation. So let me just go back through that a little bit and give an example. There was a disease we were working on recently where there are about 18 different databases of transcriptomic data. When our curators went through those, only 3 of those were of a quality that was actually useful for trying to make predictions. So it's really critical that we assess these databases very, very carefully. Once we've done that, our algorithms give us output that look just like these diagrams here, that then expert drug developers like myself and others in the team can go through and begin to make sense of whether there are some predictions there that could be useful for the patients. We call our system Repurpose Rank. It's a system that has several parallel methods that come together. So we assess all the methods both independently and together to rank the top drugs that may be useful in the disease that we're trying to treat. So the way that the omics matching part works, this is just one of our methods, is if you look at the top bars there that are pointing upwards, overexpressed. So it may be that in a particular disease, some genes are being overexpressed compared with somebody without the disease, and that's represented by those 3 bars, or some may be underexpressed, that's being represented by the 3 bars pointing downwards. So that's a disease signature. Obviously it's a lot more complex than just 6 bars, but that sort of illustrates what it's the basic principle. So what we're looking for is a drug that reverses that phenotype. In other words, it balances the disease by making the genes which are overexpressed now could be underexpressed, or at least brought back to a normal level by the drug. So that you see with the pink bars are now pointing in the opposite direction. Put the drug together with the disease, you basically produce a neutralization of the overexpressed genes. Of course, it will never be perfect, and it may look a little bit like the diagram that says therapeutic result, but it will bring it more back to normal than the patient is experiencing. So very simple concept, but it does seem to work. And an example we've worked on is CDKL5 syndrome, where we went through the whole process last year in just a few months of data curation, analyzing the data, selecting the potential drugs of interest, and then took them into experimental assays in Italy. There was only one laboratory in the world that has suitable assays for this disease. We picked out a drug called tianeptine, which is an antidepressant discovered in 1968. A drug with quite interesting mechanisms in depression, quite different from most of the highly used antidepressants. And we found that in the assays run in Italy, our drug predictions were correct. The drug did restore the profile back to normal. In this case, this is what's called the GluR2 receptor, and it restored the expression of that receptor back to the normal level. So we have other methods in addition to omics. Guilt by association means that we're looking for, probably nearest neighbor would be a good description of this. We're looking for patterns in either drugs or particularly their chemical structures, their targets and the side effects, and we're matching that to phenotypic response, disease ontology and gene expression. So pattern recognition that points us to drugs that may have the same effect as the drug that's showing a maybe weak activity in a disease, a drug that could have a better effect. The classic way of doing this is really shown in the right-hand diagram there, where we have a disease, one drug works on it, another drug working on the same target will then work in the same disease. What we're doing is much more complicated than that, much more sophisticated, using the advanced methods shown on the left there. We also have literature mining methods, which I mentioned earlier, based on natural language processing that allow us to select from the mass of scientific literature, medical literature, drugs that may be applicable for a disease, even if it isn't obvious just from the biochemical methods. And drug network analysis, which I think is one of our most interesting approaches, where this is now based on drugs, grouping drugs that have a similar transcriptomic response. Not just grouping drugs that have a similar biochemical mechanism, which is what we've done classically over the last 30, 40 years, but now we're looking at the phenotypic response of a drug on the genome, the transcriptome, and grouping drugs that way. And then we find groupings of drugs, nearest neighbors, that are not obvious by classical biochemical analysis. So again, it's pointing us to potential therapies that could be tried for rare diseases. Okay, so that brings me to the end of my presentation, and I'll open for questions. As I said, I have with me Tim Gilliams, who is the CEO, and he understands the programming behind this much better than I do. So if you have very detailed questions, I'm going to pass them to Tim. And we have Ramilla, who's our commercial head, also a very good scientist herself, I must say. So I've got two people here to help me with questions.
Okay, great. Thank you, Dave, for a very interesting and useful presentation. So as Dave just said, we're going to open up the floor to questions. Well, first we have a comment from Alan Thomas saying good work to Helix, by the way. I think he found the presentation really useful. So Ramilla says thank you, Alan. Cool. So the first question we have is again from Alan Thomas for Rick. He says, do the bars in the graph, I think the one about with the brown and bars and the green line, do they compare with the UK QALY from Public Health?
So I guess you're talking there about the new rules that are coming in for NICE, so the need for a threshold in cost for drugs. So that'll be 100,000 per patient per year on that graph, which would then be allowed for reimbursement without the further need for investigation. On the graph that I showed looking at drug development prices, that wouldn't correspond there. So that was looking specifically at the cost of development, of research development for drugs and the number of entities that have been produced. I'm trying to think if there was another slide that looked at drug costs. Generally speaking, though, from what I know, there's a number of drugs that have gone through recently and have been approved by NICE that have a cost that is higher than that new threshold. I think it's around the 25%, if I'm not mistaken. So yeah, that's certainly an area to look at. In terms of quality of life measures, so measures that are trying to assess health economic input along with its impact on quality, that isn't something we've yet looked at Find a Cure in our health economic analyses for the social impact bond. It's something we could look at and develop. With that, you need a lot more data on the current impact of a disease on the patients themselves, and it's certainly an area that patient groups can actually contribute quite strongly to help understand the natural history and progression of a condition, and also its symptoms and effects on patients. The reason we didn't look at quality of life is firstly because it's another layer of complexity, but secondly what we wanted to do was really show a very simple argument to NHS and other people involved. We don't need to incorporate necessarily the benefit to patient into our calculations to make it look viable. We can just look at the economics and so show we are saving money through improving the patient healthcare. So in our models, all of the savings are a result of a reduced need to deliver treatment or services or interventions to the patients. So it's really implicit that we're improving the health of the patient, and at the same time we're delivering those reduced levels of spending into the health service. So to some level, not using that quality is almost more compelling. We don't need to do that, but actually I think in terms of long-term analysis of rare diseases, there needs to be a lot more focus on looking at qualities and quality of life assessments to understand really their impact on patients and to really show the value of many of the new treatments that are coming out from the pharmaceutical industry, which in many cases are fantastic and exceptional and really do need to be delivered to the patients. So I hope that answers your question, Alan, and if not, I'm sure you'll let us know very shortly.
Great, thank you, Rick. So we've had another question come in, and by the way, if anyone does have any more questions, please do send them in. We'll do our best to answer them. So this is from Michelle Connor, and she says, is it common in rare diseases for the NHS to start to prescribe or use drugs before full clinical trials are complete?
Good question. So off-label prescription, which is prescription that's by a specific clinician, is actually very common, particularly in certain areas. So in pediatrics, in mental health, there's a lot of situations where drugs are prescribed off-label for a condition that they are not officially licensed for. There are also many instances where drugs might be used at an earlier stage in their development through compassionate use. So if in a clinical trial a drug is showing a lot of promise and benefits to patient groups, it might be deemed entirely appropriate that patients should have access to that drug in advance of its full authorization. So there are a lot of instances where that does happen. In the rare disease field, off-label prescription is also relatively common because there are situations simply where clinicians have an option of doing nothing or trying something that can potentially benefit patients. So there might be cases where there are case studies that are done on an off-label used drug to patient groups to see how it can affect things. But in many cases, those kind of studies just aren't done, and we don't really know the level of this off-label use or the success and benefit of the off-label use. In terms of something more systematic, there are instances in highly specialised services. So these are specialist centres that are set up around the UK to treat specific rare conditions where a drug might be prescribed on an off-label basis almost as a standard to a specific patient population. And in almost all of those conditions, it will be something that's very carefully monitored and assessed and used to build up evidence on the effect of that condition. Those aren't so common, but it's not something the NHS are particularly against. But if you're talking about a condition where there is no other treatment available or licensed, so that's something we're looking at working in and developing. And I think in any case when we're talking about off-label use through the social impact bond model, we really want to focus on the idea that what we're trying to do is provide more evidence for its use and put it in a better position than that kind of random ad hoc off-label use that you might see in some cases. In many cases, the patients aren't even aware that they're being prescribed a drug off-label, and we're hoping to set up such systems whereby that off-label use will be monitored and used to build a more compelling data set to show the efficacy and effect of the condition. And I think that's something that we are very focused on. I don't know if anyone else in the room wants to say anything about that.
Just add a couple of points. I mean, Rick's answer was very good. I think the big weakness here is that there aren't databases being kept. So there's a loss to humanity when evidence is being collected for or against a drug. Another factor to consider is that a drug may be safe in the general population, but in a rare disease, there may be a genetic change that could make that drug unsafe. I think that's relatively uncommon, but there is some risk there, so that needs to be remembered as well. But the physician should monitor very carefully what's happening. Ideally, a physician should start with a lower dose than the expected dose and build up, just in case those side effects happen, but may not always have the pills to do that.
Just to add that we just recently had a conversation last week with a charity, and they did mention that their biggest hurdle was to get GPs or physicians to prescribe off-label compounds or drugs because they were at risk of their own career without sufficient data available. So there's many factors, I think, with regards to that already covered.
Yeah.
Okay, great. Thank you, guys. So this is a question probably more for Dave and his colleagues. Which approach would you say is best for, say, a patient group that has really limited resources and funding? Which would you recommend as a starting point?
Well, the important thing is to assess what's known about the disease and work with people who are capable of making that assessment. It could be there just isn't enough information available to make progress at the present time. What we can do is help advise what information would make a difference that could help us make progress. That's probably the most important first step, to work with people who really understand drug development. Academics may understand a small part of the whole process, and they may be very good on particular mechanisms or a particular disease, but also please engage people who understand the whole drug and R&D process, and they can advise just how far away we could be from a treatment and what needs doing.
And so this is Tim from Helix speaking. So we would also strongly recommend to talk to Find a Cure, because they are helping patient groups and charities, as you already know, becoming ready and contribute to the drug discovery process and become empowered. So there are a number of things and resources available so that every patient group doesn't reinvent the wheel and goes through the same struggles that other patient groups may have already solved before. So we would strongly recommend that.
Thanks, Tim. I will just chip in a little bit on that as well. I think Dave's advice is actually really good. Getting a good understanding of where things are in your condition is a great starting point to understand how you're going to forward with your research. It's very easy to be able to launch into a given project, but knowing how that's going to lead ultimately to some patient benefit is key. So if you can initially look into getting a good oversight and understanding of where things are, that can help you as patient groups or to understand where next to go. So part of that will be in setting up a good scientific advisory board who can help you in that review process. Part of that will be making contacts, talking to different people in industry, and hopefully, some of the work we're trying to do in our networking events, our conferences, or our online portal will give you some ideas on where you can build those contacts. And it's giving yourself a nice base to start from and understand how you can move forward, and then building the connections with people who can help guide you along the pathway. That's probably the best way to move.
Okay, great, thank you. So I have another question come in about the SIB. So do you need a complete health economic model, and how much does it cost to develop one?
Good question. Do you need one to submit to the open call? No. Is it better if you've got a good understanding of health economics? Of course, but we kind of fully understand that this is not something that's done commonly in rare diseases. There are very few health economic models out there for rare diseases. So that's why in our proof of concept study, we really focused on health economics and developing models for our 3 conditions. At Find a Cure, we've got some strong collaborations in this area, and one of the things we're very keen to look at doing with groups we decide to work with through the open call is to help develop some health economic modelling to really robustly assess the current cost of the NHS or maybe other healthcare providers if that's appropriate, and how the proposed repurposing project can be impacting NHS budgets. So That's something that we feel we can help with, and if it's something you're interested in developing independently, please do get in touch with me, and I can help you with some contacts to do that. How much does it cost? It will really depend on who you're working with and the scope of the research. You will probably need to develop a collaboration with a pharmaceutical consultancy company, someone who's got experience in health economic modelling. It's good to have contacts then with clinicians who are delivering treatments as well, because you don't want to build a model that is based on a treatment pathway that isn't representative in your area. But with that, in the order of a couple of tens of thousands would definitely deliver it, maybe much less than that depending on how you can work. The important thing to remember though is many of these agencies are very open to working with charities on a pro bono basis, and with the right project and openness on both sides and understanding, I think these projects can be delivered free with work given from the charity itself.
Great, thank you, Rick. So we've got another question in, which is, assuming that someone has identified a generic drug with anecdotal evidence in a rare disease, could you please describe the time required for a typical phase 1 or phase 2 trials in rare diseases? How many patients are required for each, and any ballpark costs? At what point would one apply for an IP? I can break that up if that's quite a long question. Do you want to start with describe the time required for a typical phase 1 or phase 2 trial in rare disease?
So it's actually going to vary from disease to disease. The real issue usually is the time it takes to recruit the patients. And for rare diseases where patients are scattered over probably quite a wide area, that will be limiting. So that will tend to extend the normal timelines and possibly the costs. For an ordinary drug, for a large patient population, phase 1 typically takes 1 year, and phase 2 takes 2 years, and phase 3 takes 3 years. They're the kind of average figures. That depends on an organization that has the people in place and the contacts to meet those timelines. But that's about the fastest it can be done. I think, though, for rare diseases, they're going to be pluses and minuses. I think, though, as I said, patient recruitment might be slower. On the other hand, it may be possible to keep costs lower because it's been done for compassionate use rather than commercial uses. So probably longer timelines, but lower costs, I would guess.
Yeah, I'd echo that generally. I think just a point to make is it can be very hard in recruitment for rare diseases, and that's something that you hear about a lot when you're touring around, going to conferences. But obviously, that's a space that patient groups can play a huge role in. So if patient groups have those contacts in place for other patients around the country or around the continent or around the world, that can really help to spread the word about new trials and potentially speed up recruitment. It can certainly give access to a good base for some of the earlier preclinical work. It may be that when you're coming to a phase 2 or maybe a pivotal phase 2 study, when you're trying to do everything in one, it might take a little bit longer to deliver the recruitment itself. But there are a number of examples where recruitment has been very successfully expedited through patient groups. So the AKU Society are a great example of that. AKU is a hugely rare disease. I think they know of only 1,000 people in the world who've got it based on their work over the past few years, but they managed to recruit, I think, around 140 patients for a phase 3 trial inside a year, and that was excluding a huge chunk of the UK population in that example. So something that's really hard to deliver can be accelerated through that work.
David here again, just to add one more thing. Often the timelines for developing a drug are driven mostly by the number of patients that regulatory authorities require to build a safety database. So for instance, with Viagra, the drug was developed at very low cost and very fast because the efficacy was just so obvious and stunning in most patients. But the limitation was the safety database. We had to have it in 1,000 patients. I think it was actually 900 they accepted in the end. And that took us time to do it. So it added a year or two to what would have been the timeline otherwise. I think for rare diseases, and particularly repurposing, that won't be the case. Because remember, we're using a drug which has got a big safety database already, even bigger than a drug that's being newly launched. Provided nothing unexpected is showing in patients, the need to build that safety database, I think, will be much lower, and so the timelines may be helped by that.
Okay, great, thank you. So the kind of second part of that kind of longer question was about any sort of ballpark cost, and at what point would one apply for IP?
Okay, cost and IP. That's a very difficult question, and it depends hugely on how you're planning to go about the project. So on IP, it would depend on what intellectual property might be available. So if you're doing a repurposing project and you're looking at a generic drug, the intellectual property, I suppose, would likely be in the way you want to, the mode of use, as you mentioned earlier, how you're going to. Oh yeah, how you're going to deliver that. Which population you deliver that drug to, and potentially whether you might be changing the dose or the method of delivery of the drug. So if you're working to change the way it's delivered, maybe switching it from a tablet to an injection, or vice versa, or changing the dose itself to something higher than is currently licensed, that would give you intellectual property. And in that situation, you would apply for that as soon as you are able to protect it, really, and you've got the evidence that there's value in doing so. With IP, it's always about getting in as early as you can to make sure it's safe, and then you can allow your publication. But you really want to do that also in a stage when you believe you can still benefit from the IP down the line. Costs will depend on the type of trials you're running, the scale of them. If you're looking at doing something in a very strict back academic setting, we've heard of phase 2 trials being delivered in the tens of thousands, which is a very, very low level. This is unlikely, and there are many more that are much, much, much higher. Hundreds of thousands, millions, you can get into billions when you're talking about a full-on pharma development. So it's pretty hard to give a number. I would expect you're always going to be wanting high hundreds of thousands in an ideal world for a full route, even on an academic plane, but we think there are ways to strip things back. So Cures Within Reach, for example, tend to give grants for repurposing projects in phase 2 studies only, leading to an off-label prescription, and they talk about figures in the $150,000 to $250,000 range. So it's possible there, but you're not leading to a fully marketed drug, and I'm sure there'll be very different perspectives on what you're aiming at and what you should go for. But I'm going to let the experienced man, Dave, to my right, give his opinion.
Okay, firstly on IP costs. I'm filing patents all the time, and the costs are painful. It depends what your intention is. I look at patents these days, since I don't work in big pharma anymore, as really freedom to operate. In other words, you're giving yourself the freedom to use the drug in the future, not stopping other people using it necessarily. In which case, if you really if you're a UK charity and you want to have the drug available for UK patients, you only file a UK patent, which isn't particularly expensive. It's a few hundred, or probably it depends on the agent you use. Many agents will give cheaper prices for charities, so you can negotiate that. The initial filing costs aren't that great, but when you go to PCT, as it's called, which is the sort of worldwide filing, what you end up with is translation costs into other languages. And it's actually the translation that's the major cost, especially when you get to Japan and China and other countries. And so you really have a choice, just how wide you want to go. If you're going to go globally, when you get to PCT, you're going to be running into £30,000–40,000 costs at that stage. But if you just want to do it in your local country, the costs are actually quite low in your own language. In terms of cost for clinical trials, what really drives those costs up are the failures. Most drugs fail. They say that about 1 in 10 that enters clinical trials actually succeeds. I think the figure is actually much worse than that, but that is the best case, 1 in 10. For a rare disease, and particularly the way we're going about repurposing, we should be in much better shape, because we're looking at the genotypic response in individual patients, matching the drug to it. So we'll have a diagnostic for patients of whether the drug could work. And what's often failing with most drug development programs is we just don't have a diagnostic that predicts which patients would work. Therefore, in the trials, you would only include those, you would basically type each individual patient that's going into the trial, take a cell sample, see if the drug works on it in the way predicted in the initial studies, exclude those that it doesn't work, and therefore the success rate should be much higher, so that the patient numbers required in each trial should be much lower, the cost should be much lower. So I think that there's an enormous advantage, I think, with repurposing, particularly using genomic approaches.
Great, thank you. So I have another question that's come in. If a rare disease has a very small cohort in the UK, then will it not be quite disadvantaged in the open call because its cost to the NHS is lower?
That fully depends on the cost of treatment and managing the symptoms, really. The model certainly works best when you have large potential savings to the NHS. And there are two ways that works. That's either a high individual per patient cost saving. So if you're looking at a condition where you have frequent visits to intensive care, really high costs from surgeries, a lot of time interacting with the NHS, that then there's a potential that a very rare disease, an ultra rare disease, could really deliver significant savings into the condition. Equally true if you've got a more common rare disease, something where you're talking about thousands of patients in the UK, you need much smaller per patient savings to work in this. We are not claiming it's a perfect model, and there are definitely going to be cases where you have ultra rare conditions that are not really having that much contact with the NHS, and in those cases this model isn't quite delivering yet for those conditions. What we would hope to say is, if we can make it successful, if we can show that we're delivering treatments and savings through 1 or 2 rounds of our portfolio approach, then we can begin to take on some of these conditions into it, with the aim of our success payments, our excess funding, paying for that research without the expectation of returns into the model. We can begin to carry some treatments in there. But it is admittedly not perfect for everyone, but we're hoping it can improve the situation for many more. I'm just going to pass over to Tim now.
And I think it's worth mentioning that I think the power of the consortium we've put together here makes that with Cure Accelerator and Cures Within Reach, we could also access the necessary philanthropic funding to validate and get some of those pivotal clinical trials off the ground, even if for the social impact bond, it wouldn't fit exactly the criteria.
Yeah, that's true. Thanks, Tim. There's lots of other routes we can look at in those conditions, and we're keen to do that for the open call.
Great, thank you. So we're out of questions for now, so while we just wait for a few more to come in, I'm just going to mention a couple of other things that we've got coming up at Find a Cure. So firstly is a webinar titled Medical Research Explained. This will feature 2 guest presentations who will introduce the different stages involved in medical research and explore how patient groups and other advocates can find the most appropriate research to sort of get behind and support. And then on the 4th of May, we'll be heading over to Cardiff for the next in our series of networking events. It'll be the Cardiff Rare Disease Showcase, and it'll take place in the evening at the Life Sciences Hub in Cardiff. The aim is to bring together the rare disease community, so all patients, patient groups, charities, researchers, clinicians, industry representatives are welcome. Anybody at all with an interest in rare diseases. It will feature 2 case study presentations. Delegates are welcome to submit lightning talk suggestions and might have the opportunity to present. There'll be plenty of networking and lots of refreshments and canapes as well. So we'd love to see you there. Oh, and it's free to attend for patients and patient groups as well. So
just want to quickly add on the Cardiff Showcase. We'll be accepting for 2 more weeks proposals for talks for the event. So if you're interested in speaking at that event and giving a lightning talk, if you register within the next 2 weeks, we'll be able to consider that. And after that stage, we'll be selecting our program for the evening. So please do check out and register now. It's a great opportunity to share some of the stories and research in your condition.
Cool, thank you, Rick. So we've got another question in. Are there any clinical CROs you are aware of who specialise in rare diseases?
Good question. There are a number that are working and have rare disease divisions. So there's a couple of different ones I've got some knowledge of. I think Tim has an example to share.
Yes, we've been contacted recently by a CRO called CTI, who seems to have a strong focus and interest in repurposing and rare diseases. The challenge of rare diseases is that often you don't have actually all the assays available, all of the data available, so there are a lot of gaps there, but I think there is an increasing interest for that, and I think CTI is one example, but there must be quite a few more.
Cool, thank you, Tim. So no more questions have come in, so I think unless anyone else has anything to say, cool. We'll wrap up. So thank you very much, everybody who's stayed with us and listened to the webinar today. As you can see, we've got contact details for our panelists here. So Rick's email address is there, and also Ramila's, and Dave's was earlier on in the presentation. You can follow both organisations on Twitter, and our web addresses are there as well. If you wanted to go straight to find out either about drug repurposing or open court with Find a Cure, after the findacure.org.uk, you can do /open-call or drug-repurposing, and that'll take you straight to those pages. So yes, thank you very much for joining us, and if you do want to get in touch afterwards, then please do. We'll be very happy to hear from anybody.